Hello, students. Good to have our second week here at Midland College, and hopefully all are getting along pretty well. This is going to be our second week, and we're going over the topic of macromolecules. This is still in our Unit 1, which is over cell biology. And so, with that, we're going to go ahead and get started. So, this lab covers macromolecules. So what are they? Well, they're large organic molecules, right? And so organic molecules, right? Those are basically compounds that contain a carbon and a hydrogen atom. And these are all held together by covalent bonds. These can appear in chains or in rings, okay? Inorganic molecules, however, are compounds that do not contain a carbon and a hydrogen together at the same time. And so these are going to be, different inorganic molecules such as water, oxygen, oxygen gas, nitrogen gas, and our salts. And so here we have our most basic kind of idea to get you familiar with organic molecules. But here we have, just a simple glucose molecule, right? And these are different ways that you can draw your glucose molecules. I personally prefer this way over here, but y'all aren't going to be doing too much drawing of molecules, but we will be looking at them to understand how they form and how they break. So that's going to be today's assignment. So here's an example of an inorganic molecule, and as you can see, right, this is H2O, right? And so these form when one is more positive and one is more negative, and so that is how that attraction is formed. Okay, so we have this negatively charged oxygen molecule with these positively charged hydrogens, and they're forming that ionic bond. But let's take a closer look at things, right? So when we're talking about covalent bonds, what exactly are those, right? These are bonds that are formed from the sharing of valence electrons, right? And so these are usually two nonmetals that are forming a covalent bond. And so let's look at this over here. Okay, so we have a hydrogen and a hydrogen, right? And hydrogens offer one valence electron to share. And so you can have this hydrogen gas form from just two each sharing an electron to form that bond, right? Can each share that valence electron. Here we have some oxygen, right? And so oxygen can go ahead and form a double bond with itself, right? It has two of these little covalent bonds right here from their two different valence electrons that were available. Carbon is particularly amazing in that carbon always offers, right, 4 different valence electrons. So carbon's always going to be able to form 4 different covalent bonds just because it has that fourth one available. So here we have one of either carbon or hydrogen, it doesn't really matter, but one of them is offering a valence electron, and hydrogen is able to form with that. So typically carbon always is attached to a couple of hydrogen atoms right there in covalent bonds. And so right here, this one is showing you a non-polar water molecule where they're actually sharing these electrons with oxygen, right? But let's talk about ionic bonding, okay? Ionic bonding is going to form, right, from oppositely charged ions. And so sometimes it would be something like a non-metal and a metal. Over here we have the formation of, sodium chloride, right? A salt, right? And so you actually have, right, sodium giving its electron out to the chlorine atom, right? And so when that happens, this becomes our sodium ion, or it becomes an ion, and it becomes positively charged because it no longer has that extra negatively charged electron. It gave it over here to the chloride atom. And so we have this negative attraction and this positive attraction to one another, and that's how those bonds are formed. And so with that in mind, let's go ahead and move into the 4 different types of organic molecules that you can make based on these different types of bonding patterns. So what we're going to be looking at today are the 4 different types of organic molecules, which is carbohydrates, proteins, lipids, and nucleic acids. And what can these be? Well, they can be what we call polymers. And polymers are just large complex molecules. And they're formed from a bunch of smaller molecules that we call monomers. And monomers are always held together by covalent bonds. Okay, and so let's look at that. So glucose by itself is a monomer. It is just this little molecule right here of glucose. That's all it is, right? But we want to be able to combine it with other monomers to make that large polymer. And how do we do that? Well, we have this thing called dehydration synthesis. And what is dehydration, right? Dehydration means to lose water. Synthesis means to build, right? So dehydration synthesis is the process where monomers will bond to each other and they'll form polymers. And that happens because they lose a molecule of water between each pair of these monomers. So let's see what that looks like. So we want to create this polymer right here from each of these monomers. So let's look over here at oxygen. Okay, oxygen needs to get rid of this hydrogen, and then over here we need to get rid of this this OH group, right? And so to do that, well, we have to produce water, right? So that way these bonds can form between each other. Oxygen, right, as we know, it's able to have 2 bonds because it has 2 valence electrons. So once that gets spit out, it can form with this one hydrogen ion, and that produces water. And from that, this oxygen that's left behind can form a bond over here to the sucrose molecule. And so, just remember, dehydration, lose water to build a big molecule. But what if we want to break these down, right? So we've just consumed some of these these sugars, right? We've consumed them. Now we want to break them down, digestion, right? How do we do that? We do that with a process of hydrolysis, right? Hydro meaning we have water, lysis meaning to break down. So this is the process of breaking down your large polymer into a monomer by just adding the molecule of water. So it's exactly our other process but backwards. Okay? So hydrolysis happens when we put in this water molecule, and so then this bond breaks, right? And the O is going to instead, right, attract to our hydrogen. And then our little empty bond right here, this valence electron left by the carbon atom, it's gonna attach to our hydroxyl group right there, the OH. And so this is them side by side together, right? Building up our sugars. We're gonna make one very large polymer, right? We have each one polymer here. We make a monomer, and then they're gonna form a polymer, right? And then you have this large polymer, and we're gonna use water to break it down into these separate monomers. And so with that, now that we know how to build them up, let's talk about what they are. Let's talk about carbohydrates, right? So carbohydrates are sugars and starches. They're composed of one carbon molecule, two hydrogen molecules, and one oxygen molecule. So we call this the 1-to-1 ratio. That's how you find a carbohydrate, right? And so there are 3 different types of carbohydrates, right? And so we have monosaccharides, right? These are carbons or carbohydrates, right, that form very simple monomers such as sugars, glucose, and fructose, right? But then we also have disaccharides, and so these are polymers, right? But composed of two different types of sugars. So sucrose is formed from glucose and fructose. So that's a disaccharide. Polysaccharides, however, are formed from multiple different types of monomers of these. So polysaccharides are formed from multiple monosaccharides. Disaccharides are only formed from 2 monosaccharides, okay? And some examples of this is starch, glycogen, and cellulose. So once again, we're just kind of showing you your monosaccharide for glucose, your monosaccharide for fructose here. And they're going to go ahead and they're going to form together, right, by getting rid of that water, right, to form a disaccharide. Okay, so same concepts, we're just applying more specific names to them. Okay, same process, right? We're getting rid of water to form a disaccharide. Okay, but how do you get polysaccharides? Well, these form from multiple of these little monosaccharides. Okay, we've got a bunch all linked together, and these are our starches. See? So we can usually find these in plants, right? And so plants, what are they composed of? They're composed of cellulose. Cellulose is used for structure. So cellulose is an incredibly strong molecule, right? It's incredibly strong. It's really hard to break down. We can't digest cellulose. That's why we eat, like stuff like spinach and kale and stuff. We're adding roughage to our diet because we ourselves, we can't really digest that at all. We can't digest cell walls of the plants. It's just too hard for our bodies to break down. So there's just some more up close images of these different cellulose fibers that come from the cross-linking of these cellulose molecules. Another instance of a carbohydrate, right, is chitin. And what is chitin? Well, it's a molecule, right, or a polysaccharide that we find within cell walls of different fungi and in the exoskeletons of arthropods. Okay, and so these are incredibly strong cell walls from these different organisms, or well, in the fungi, and then incredibly strong material on the exoskeletons of arthropods. They're hardy and they've led to their survival because they're that strong. Now, you're also able to reduce or have sugars reduce other groups, right? And so some of those can actually be monosaccharides and sometimes some disaccharides. So what does that mean? Well, they have the ability, right, to give electrons to other molecules, right? And they're able to reduce other molecules. So when you give an electron to something else, that that atom is then reduced. Not atom, I'm sorry. That other molecule is reduced, okay? And when they do that, they free up a carbonyl group, right? Like I had said, carbons have 4 different valence electron uh, positions that they're able to go ahead and have bonding to. But when a sugar reduces another molecule, it can free up that carbonyl group. So what are some of the functions of carbohydrates and why do we even look at them? Well, they're important for energy, right? They're an energy source for our cells to produce, ATP and all that good stuff. So, glucose, right? And then in plants, they're the ability to store this energy, and that's in the form of starch, okay? In us, it's glycogen. And it's also a structural material, right? So like we had discussed, the plant cell walls, its structure, it's keeping it nice and strong and sturdy. And how we can test for these different types of macromolecules is by using what we call a Benedict's test. And so we can test, right, to see if a sugar is reducing another molecule. And that happens, right, when we do this Benedict's test, and you'll get to see a very obvious color change, right? And so if it's orange or green, that means it's a positive test result, meaning that the sugar does reduce the molecule, okay? And we're going to be doing this in lab, so uh, keep note of these, okay? Another test you can do to check for a different type of carbohydrate would be the starch test using iodine, right? And iodine is a very um, this color can stain a very dark purple, almost black color when it comes in contact with the starch. And so it's very easy to tell using iodine when it's on a starch because it will do a very vivid color change. So on from our carbohydrates, we're going to go ahead and talk about lipids. What are lipids? These are our fats. These are our oils, our waxes, steroids, and phospholipids. Okay? And so how are lipids formed? Okay? Well, they're formed by that little process we learned, the dehydration synthesis, right? We're going to lose H2O to form a bond. So right here, this little H is lost, and this hydroxyl group is lost, right? We lose those on each. They're gone. And now we're left with 3 molecules of water, and we have a nice bond, right, of 3. And this is for a very particular type of a molecule, the triglycerides, right? You have a triglyceride, and that's how you're able to form a lipid. And just here's that same example shown a different way. So, like we had said, lipids are different steroids, right? So we can have cholesterol and then testosterone. These are very important in the functions of our body. In males, this helps to produce, different growths and different hair, all kinds of processes in the body. And so when we look at our lipids, right, they have a very, very obvious morphology, right? They have a very obvious makeup of their body. And so they have this hydrophilic head, right? Right over here. So we have our polar head, meaning it's charged, right? It's polar. And then we have our hydrophobic fatty acid tail. Okay, so meaning it doesn't like water. Okay, hydrophobic. Hydrophilic, it can do fine with water, right? And then we just have our glycerol backbone. Okay, where do we see this? Let's see if I have a draw option. I do. Where do we see this? See this in our cell membranes, right? So, pardon the bad drawing, but here is what we have, right? That's a little lipid, right? And we have a phospholipid bilayer in our cell membrane. That's what our cell membrane is composed of, right? We have these two little opposing lipid bodies that form all of our cell membrane, right? So we have our hydrophilic head and then our hydrophobic tails, and those line your entire body. So imagine that these two lines are filled all the way with just these little molecules, and that's important to know. So let's move on. So lipids, right? They're soluble in nonpolar solvents. So, you know, just means that they're able to be, broken down into either ether or acetone. However, they're insoluble in polar solvents, such as water. So, lipids are fats and oils and stuff. So, what does oil look like when you put it in water? It doesn't mix ever. Okay, and so that's just how you're able to tell what it is. Okay, and like we had discussed, right, polar molecules are just molecules that show a charge. So in lab, we're going to be doing these different tests for the lipids, right? We're going to have our Sedan 4 test, right? And so this is a nonpolar red dye stain. And what it will do is it'll stain a lipid layer, okay? And so it'll be a bright red if it is lipid, okay? If it's anything else, it's not going to stain it. Easiest one to do is a grease spot test, okay? So you can see if it's leaving a greasy spot in a piece of paper, and that's how you can tell if it's a lipid, If it's fatty, greasy, it's going to leave a spot. Why are lipids important? What do they do? Energy storage in animals, right? And in some plants sometimes. Think of, I guess, cashews. Yeah, cashews and avocados. Those are the only things that really come to mind when I think of plants having fatty material in them. So they do occur, but they're not too often, right? Cashews are good fats, I think. And so same with avocados, right? They're those natural fats. But for us, energy storage in the form of fat. And there are also waterproof coverings on leaves and stems of plants, right? And so this will help prevent the plants from desiccating or, overly getting saturated with water as well. And so it's also a common component of plasma membranes of our cells, like I had said earlier, right? So those just are what make up our membranes of our cells. And then also thermal insulation, right? You look at a polar bear; they never look cold out in out in the cold, right? They're just fine. They're insulated by their many lipid layers. They're also for cushioning and shock on absorbing, for our organs and our bones and our muscles. So without that, we'd have no no lubrication for our bones, when they grind against each other or anything like that. So lipids are incredibly important. And let's move on to proteins. So what are proteins? These are macromolecules composed of amino acids held together by peptide bonds. And so we have 20 different amino acids in living things, okay? And so this is just kind of showing you the chemical makeup of an amino acid, right? And so you have your amine group, right? Your amino group, okay, which is just a nitrogen and 2 hydrogens, right? Then you have sometimes a variable group out here. It can be another molecule attached. We have our hydrogen, we have our 2 bonds formed here, 2 bonds formed here. And then we have our carboxylic acid group, right? And so that's when we have our carbon, right, double bonded to an oxygen, but also to that hydroxyl group. So this is, typical structure of amino acid. And so, as we had said, amino acids, right, they basically compose, or they build up a protein. And so, what are some of our amino acids, right? Well, here we have something like glutamic acid, right? We have arginine, phenylene, leucine, cysteine, and these all form, right, to produce our proteins. Okay? And once again, how do we make that? Well, with dehydration synthesis, right? That's getting rid of the water to form a bond. Okay? And so these amino acids form this polypeptide chain right here. And so, once again, we're doing that peptide bond formation by losing water. And so, when we have the formation of a polypeptide chain, right, we're able to produce a protein molecule, right? So kind of back to the monomer idea, right? So all these little amino acids are basically little monomers, right? Producing a larger protein, right? And so this molecule is very large. It's a protein. And we have a lot of different protein structures. And you'll get more into this if you ever decide to take like a chemistry class or go further into biology. But we have secondary proteins and tertiary proteins and quaternary proteins, and they have different formations. Okay, and so we're gonna not really focus on that today, but why are proteins important? Why do we study them? Why do we need them for life? Well, they're necessary. They're necessary for growth, okay? They're necessary for repair, for components of your hair, your skin, your nails, and your muscles. They also are, important enzymes that we use. Hemoglobin, right? That transports oxygen in our blood, right? And so these are all very important things that we need from proteins. And you can get protein just from, different sources. We get them, by eating them. So you can drink some milk, you can eat some egg whites, meat, peas, beans, nuts, legumes, all that good stuff. And so how do we test if something is a protein? Well, we do a burette test for protein. And so what will happen is we'll get a violet purple color, and when it becomes that color, you know that it is a protein. Okay? Violet purple, protein. And so how this happens, the burette reagent, it just reacts with the peptide bond that hold the amino acids together in the proteins, and it's able to dye that, and we're able to see it. Okay, and so from proteins, right, we're going to move on into nucleic acids, which of course, when you hear nucleic acid, you immediately think of stuff like DNA, right? And so DNA, right, is composed of a phosphate group, a simple sugar, and then a base, right? And so for RNA, right, it's only composed of ribose. But in DNA, we have deoxyribose. Okay, so look at those. And let's move on. Okay, so you can form a nucleotide chain. And you can do that by having, right, our different little phosphate groups, right? Can all have them up there. So we have our little nucleotides themselves, and they're able to form a chain. And just like we have discussed before, this is done through some hydrogen bonding, right? And so A to T's, they'll always have 2 hydrogen bonds. C to G's will have 3 hydrogen bonds. And these are what form, you know, all of life. Okay? And so this is kind of just showing you, right, how DNA can be synthesized, right, by using parental double helix, right, the DNA strand, right, this is your parental strand, and it'll split, and you can form new strands by using the old strand as a template. Now, I thought this was cute. It was already included in the PowerPoint, but how do you remember which one's linked to which? Well, T's go to A's and G's go to C's. So it's easy to remember if you say Trans Am. Great car. Wow. They even have little T to A, G to C. And, you know, those aren't the only nucleic acids that we have. We can also have cyclic adenosine, right? And this adenosine monophosphate, right? So this is cyclic AMP. And this is incredibly important. If you ever take a cell bio class, you get more into it. But it's basically important for intracellular communication, right? Then we have adenosine triphosphate, ATP, right? We need that. It's our energy carrier. And then we also have coenzymes, right? And those help to be active in cellular metabolism. So very, very important. And so with that, right, we're going to be conducting a series of experiments in lab. And so I hope you all look over your lab instructions before we go into lab tomorrow. Thank you for listening, and I can't wait to get to the actual experimental part of this lab. We'll be conducting a bunch of these different tests on a bunch of different, samples in the lab. So, see you then. Bye.